All right, so I'll go ahead and start. Professor Andrea Baccarelli is our first speaker. He is the Leon Hess Professor and Chair at the Department of Environmental Health Sciences, Mailman School of Public Health at Columbia University in New York. Professor Baccarelli has contributed to the development of international best practices for air pollution control, developed with multiple agencies worldwide. His findings served as the basis for the U.S. Environmental Protection Agency's decision to enforce stricter guidelines for human exposure. He is known for his willingness to collaborate across all disciplines. In 2020, Professor Baccarelli was elected to be a member of the National Academy of Medicine. His research investigates molecular mechanisms as pathways linking environmental exposures to human disease, including epigenomics, transcriptomics, extracellular vesicles, and small non-coding RNAs, mitochondrial DNA, and microbiome. Please, Professor Baccarelli, welcome.
Thank you so much. And I guess I need to share my own screen, correct, here? Do that.
Not yet.
Can you see me? Can you see that? Fantastic. So today I would like to start by, so it's a great pleasure to be here, to be presenting online. Sorry I cannot be there in person, but great I can do this over Zoom. First of all, I would like to discuss what epigenetics is, and also particularly why we are interested in it. So epigenetics is usually defined as the programming or gene expression.
Hello, sorry, Professor Baccarelli, they're telling me that you can share now. You should be able to share your slides.
Oh, sorry, I didn't do that. 1 second.
Yeah, we can see that. That's perfect. Thank you.
Sorry, I didn't push the button in the end.
Thank you.
Sorry about that. So epigenetics is about the programming or gene expression that does not depend on the DNA code, and it's stable because it can be propagated from cell to cell through cell division. So it goes from the parent cells onto the daughters. Specifically, this has been also implicated in transgenerational inheritance, and I'll talk a little bit about this. A couple of characteristics of epigenetic programming that I want us to keep in mind is that they are modifiable. Therefore, these are programs that can change, and in particular, they are associated with genes that are active or poised to be activated. So being a program can be about the future, like our programs for the weekend or for the holidays. I mean, they are in the future. They're not happening now. They're not happening yet. So this is particularly important because we have been working on the epigenome, hoping to find not just the current health states, but also predict future disease, health states and disease. And I would like to give you an example of what epigenetics is about. It's going to be a musical example. So what you see here on the left-hand side is the DNA. The DNA particularly is a code like this music score that you can read to generate a phenotype. And the phenotype will be, of course, a symphony that can be of different types. And you can imagine how the symphony can be amazing, or can be also very, very much worse depending on the situation. So the same score gives you many different phenotypes. So this is what we know happens with DNA. The same DNA can give you many different phenotypes. So, but this doesn't tell you yet where is the epigenome. In fact, you could see the epigenome if you were allowed to go on stage, look over the shoulders of each of the performers, and if you were allowed to do so, you will probably see something like this. You will still see the same score, but it will be with annotations, with markings that do not change the score, but they do change the way the score is expressed, the way the score is performed. It is something that happens in epigenetics. Epigenetics is about annotation onto the DNA. They do not change the DNA, but they do change whether the genes are on or off. So I would like to tell you also that we can use this analogy also to think about different types of markings. Some genes have markings that are in ink that are very permanent. Once they're applied, they can last years or even an entire lifetime. Some are very, very quick. They're applied quick, they can be removed quickly. And these are the ones we call impulsion. So this is particularly important for my presentation. But let me stop the analogy here, and I'll review with you that one of the epigenetic mechanisms that have been widely studied is DNA methylation. DNA methylation is the attachment of a methyl group to the DNA. These methyl groups can usually silence genes. So when a gene is methylated, the gene is off. And these are really biological programs of human cells. When a gene is, so the switching on and off of genes help us to cope with future situations, including future stressors. So I would like to review today with you what is the evidence, or at least some evidence, on the association of psychosocial stress and epigenetics, and particularly DNA methylation. I'll start from evidence from animal models. I'll review some of our human studies, and then I'll touch upon a little bit the concept of transgenerational inheritance. So let's start from evidence from animals. So this is a model that was published now almost 20 years ago. Sorry, this is a model that's much older, which is a model of maternal care in early life. So rats can be classified and spontaneously show how they take care of their pups, so their offspring, by the level of maternal care they provide to their pups. And particularly one common measure is the amount of licking and grooming that they give to their pups. And so you can see here, for instance, sorry, let me get the laser pointer here. You can see here, for instance, that there is a large variation, and this is just completely spontaneous in this level of maternal care. And particularly, it has been shown that these may change the programming of the stress systems, including the hypothalamic pituitary adrenal axis, which is a very important stress response in our bodies, both in rats and humans, which control the adrenals to secrete or not secrete cortisol. And particularly, this is particularly a system that is regulated in the brain, particularly in the hypothalamus. And what was shown in this study that was published in 2001 by Mini's group in Canada is that rats that pups that have mothers that low maternal care, low licking and grooming, low LG, had when they were adults, they had higher response to stress. So low maternal care or neglect makes pups that are wired, they're programmed to have higher responses to stress. Instead, those with higher maternal care had much lower responses to stress. And this measured with the plasma corticosterone, which is the rat's equivalent of cortisol or human cortisol. But also what became evident is that this is due, or at least associated, by having many more receptors for glucocorticoids in the brain. So the brains seem to be more sensitive to the level of cortisol, to the level of glucocorticoid, cortisol or corticosterone in the circulation, and therefore they may, if they have high maternal care, they tend to lower, they feel the cortisol they have is enough and tend to lower their output in cortisol from the adrenal, while those that have low maternal care tend to have much less here receptors, so that they don't see, their brains cannot see the level of cortisol and keeps pumping out corticosterone in the case of rats, cortisol in the case of humans. You can see here the correlation between the level of lichen and grooming and the glucocorticoid receptor in the hippocampus. And this has been shown to be regulated by DNA methylation. So you can see here, particularly these slides to the left, low maternal care rats have very high methylation of the glucocorticoid receptor in the hippocampus, as opposed to these in high lichen and grooming. So what happens during the first few days of life changes the brain permanently and wires the brain to be much more insensitive to circulating glucocorticoid, and therefore pushes the adrenals to output, to push out much more cortisol, and to have stress responses that are much more intense. And this was also shown that is really due to the early life environment. These are cross-fostering experiments that show that if you cross-foster, if you have one pup being adopted by another mom after birth, they don't change their behavior, or they change their behavior only little. So this is particularly important because this shows that the epigenome, there's early life programming, especially in the glucocorticoid receptor and the glucocorticoid pathways, that are forever. The ones that are ingrained in our brain or in our bodies, they tend to remain there forever. So I want to show you the human equivalent of this study. And this is a study of child abuse and methylation in the Black Women Health Study, which is a study in Boston, Massachusetts, in the United States. So the background here is we know that early life adversity influences risk of disease, and there are several epidemiological evidence that this might happen in humans. So our objective was to assess whether a history of child abuse is associated with long-term changes in methylation levels in blood, not in the brain, in the HPA axis. We worked with colleagues at the Black Women Health Study, in which participants, about 59,000 women, were enrolled in 1995. They had questionnaires sent every 2 years, and we started the study here in 2013. And we had, our colleagues had, this is a study we did with colleagues at Harvard and BU. They had blood collection in a subset of people, only 500, performed in 2005 and 2006 in 3 cities, Atlanta, Chicago, and New York City. We studied methylation in blood DNA of the glucocorticoid receptor. We selected 295 women out of those participants with full range of data on psychosocial measures. And we measured methylation in one specific exon of the glucocorticoid gene. This is the name of the glucocorticoid gene, which is the gene that the glucocorticoid receptor, gene, which is the gene that is sensitive to cortisol in humans. And again, we didn't do it in the brain, but in blood. What was really impressive is that when we analyzed the levels of glucocorticoid receptor methylation, depending on the reported history of child abuse. And again, these are women now in their 50s, but they reported histories of child abuse. We found a highly significant association between the glucocorticoid receptor methylation, and this the reference is no abuse. This is shown as 0 as the comparison group. And you can see that low, intermediate, and high abuse are associated with gradual increase in methylation. And this is a 2.2% increase in methylation over the reference. Also, this is the frequency of physical abuse during childhood, and this is the severity. You can see again, we find the dose response relationship from no abuse to mild, moderate, severe, and very severe. This is adjusted for age, baseline, and parental education. So this seems to show that in blood DNA, we have the same situation in humans. There is some type of programming relatively early in life, in childhood, that persisted in this case 20, 30 years. Particularly important to know, we know that abuse is an incredibly strong stressor, incredibly terrible stressor, and that creates persistent changes, may create persistent changes in someone's life. And definitely it is possible that these changes may be mediated or at least associated with changes in the regulation of the glucocorticoid receptor axis or cortisol control. Similarly, we found that another type of stressor, completely different, is associated with differences in glucocorticoid receptor methylation. These are people who work on shifts, particularly on night shifts. They're also known, workers who do nights are also known to have changes in the regulation of their hormonal pathway, particularly the stress levels. And we also found differences in glucocorticoid receptor methylation. You see, these are people who work less than 5 years on nights, 5, 10 years, and 15 years. And again, in blood, we find this amazing how indeed in animals and in humans there are these changes in glucocorticoid receptor methylation that may wire our brains and our bodies, the blood in humans, to respond differently to circulating cortisol and potentially to as a marker of heightened response to stress. So let me move now to a
Dr. Baccarelli, have you moved away from the microphone? You're much harder to hear suddenly.
How about future generation, which is an incredibly interesting area for epigenetics. So let me show you a few reflections and studies on this area. So here you have, first of all, I would like to take a second to speak about the difference between the two opposite philosophies of opposite philosophies. Can you hear me now?
Yes, thank you.
Sorry, I think you lost me for a second. So sorry. So I was mentioning I'm moving to transgenerational inheritance, and there are two different philosophies of in this area, that of Lamarck and that of Darwin. And we're very familiar with Darwin, certainly, but we also want to be familiar with Lamarck, who had contrasting philosophies. One Lamarck's example was the blacksmith. His idea was that, that we change our bodies through our life, and these changes are transmitted to the next generation. For instance, he noticed that usually the sons, not the daughters apparently, but the sons of people who have heavy occupations like blacksmiths would strengthen their arms, and the sons would also have similar muscular development when they mature. And it argued that this was transmitted biologically through the sperm from one generation to another. The other example was the example of the giraffe who he thought that giraffes would be able to lengthen their necks to reach out for foliage higher up, and they would transmit this type of phenotype to the following generation. So now we have really dismissed Lamarck straight up, but today there is evidence that Lamarck actually might not have been that wrong. Particularly, I want to show one of the many studies that show transgenerational inheritance. This is a study of parental smell experience in rats. So for rats, smell is very, very important. Not that important for humans. I mean, we use our smell, but rats depend on smell for their survival, particularly because they use it to identify threats around them, particularly predators who may be around them. So the way Diaz and Ressler, colleagues at Harvard now, performed the following experiment. They fear conditioned a male rat, so they gave them a very sweet odor, like cherry blossom, and after the odor, they gave immediately an electric shock. So after a few learning rounds, the rat becomes afraid of the odor without need of giving the shock anymore. They then mated the male rat to a female rat, and what they found is that when the progeny at the first or second generation is presented with the cherry blossom odor without being fear conditioning, they startle. They have a fear reaction, and that demonstrate that there's the possibility that what is learned by the male rat, is transmitted biologically to the following generation. They make sure that the fathers are never in touch with the offspring, and here too, to make sure there was no behavioral transmission of this phenotype. So particularly, we have a situation here where something is being learned that is transmitted to the following generation. A new behavior, an acquired behavior, transmitted to the following generation. Particularly, they identified also changes in DNA methylation in sperm in some olfactory gene. The methylation part, I have to say, is pretty correlational. There is no clear evidence that this gene they identify does exactly this. It's a little also unclear how this plays out in someone's smell and brain. But really particularly interesting. Part of the issue is that many of these studies receive a lot of attention, and these models are hard then to be replicated. I don't think this has been replicated consistently, so the jury is still out as to whether this is happening in rats, and particularly whether this is happening in humans. And the question whether this can be transmitted to human studies is still open. So I can show you a human study where I have contributed, where we showed that early life childhood abuse in men changes sperm DNA methylation. And this was published a few years ago. And we found particularly different genes that were associated with early life childhood abuse. So men who reported childhood abuse had also differences in their DNA methylation in sperm. So potentially this gets scripted into the sperm and transmitted to future generations. Also, we found that mothers who used the prenatal this is a different study, mothers who use prenatal antidepressants had changes, their children had changes in DNA methylation in blood. This study is done by Andres Cardenas, who's now an assistant professor at Stanford. So in summary, I wanted to show that DNA methylation provides traces of past exposure to stress, maybe a mechanism of disease programming. Transgenerational effects are the most intriguing aspect of epigenetic programming, and they're very hard to study in humans. It's very hard to study multiple generations in humans. The examples are still sparse. Down the line, I think we are interested, all of us, to see opportunities for research leading to primordial, primary, and secondary intervention, and the epigenome can be a readout that help us to evaluate these studies. This is all I had today. Thank you so much for your attention.
Thank you, Dr. Baccarelli. It's a very exciting field right now, a long way from alternative splicing and gene hunting back in the 90s when I was working on this. Yeah, I think, are there any questions here locally? Yes. Professor Cummings.
Yes, very interesting work. I would wish to know, you spoke about DNA methylation. If DNA methylation in relation to genetic imprinting, if there is a difference in terms of in the case of suicide, if the methylation is inherited from the mother and from their father, if there's a difference with respect to genetic imprinting, if that's quite similar.
Did you hear the question?
Yeah, I did hear the question. I think I understood almost everything. So these genes are not imprinted. So the glucocorticoid receptor gene is not imprinted. Yeah, there is plenty of studies on whether acquired risk factors impact the imprinted genes, but in fact we find that most of the signals are in genes that are not imprinted. At the same time, there is evidence from animal models that some of these effects on non-imprinted genes are gender specific. So in both at the level of exposure, so sometimes we see the colleagues see the effect if the father is exposed as opposed to the mother, and also on which of the offspring might show changes. Sometimes this is transmitted only to the male lineage, sometimes to the female lineage. And honestly, we don't know why. We don't know why this might be gender specific or sex specific in this case, I should say.
Okay, thank you. I was interested in the study with the antidepressants. Would you be able to say whether the reduction in methylation was necessarily a good thing or a bad thing, or it's just an observed effect?
So this is just an observed effect, but I need to say that most of the genome is static, never changes throughout our lifetime. The epigenome does change, and the reason why it changes most of the times is to help us. Most of the cases, an adaptive response to stressors or to changing environments. We see this, of course, with stress. The question is whether it becomes maladaptive. I'm pretty sure that the differences I showed in the glucocorticoid receptor are our body to get us to cope with stressors. The question, especially when the stress is unbearable, whether this then creates symptoms down the line. And so I think we're really talking mostly about adaptive responses that can become maladaptive.
But it doesn't seem like in general there's much to remove the methylation. You showed the mouse study where 2 generations on, they're still showing the response to the odor.
Yeah, exactly. Though that is the So we had different type of studies. For instance, one study that's really informative in this case, we did recently a large study on smoking. So we looked at people who did smoke, current smoker versus current non-smoker, and we found as many as 7,000 genes that show differences in methylation, because smoking, of course, is a heavy exposure. So all of our epigenome seems to frantically respond to smoking. When we looked at those who quit, former smokers, we found that most of these genes, most of these differences disappeared within 5 years of quitting smoking. And after 10, we found only about 200 of the 7,000 genes still there, still different. But there were some after 30 years, about 20 of those genes that still show differences. So this makes me think that when we look at the epigenome after 30 years, we do find genes that are persistently different. But if we have the capacity to look over time, actually there are a lot of genes that go away over time. So this is why I made the example of genes in pencil and genes in pen, because we see both cases in the human epigenome.
Okay. Well, thank you very much. More questions locally?
It's a long time since I did genetics, and genetics have moved forward. Is it possible to identify changes in the newborn from parents who had abused drugs, whether in smoking or cocaine or something that's more relevant to here in Guyana?
So I think the question is whether we can see changes in methylation in children, in the offspring, but the exposure is drug abuse in parents, correct? So yeah, that's a very interesting question. I'm not aware of any study of this type, but it's a great hypothesis. If no one has done this study, it would be really a great study to do, and happy to collaborate if anyone has the study population available.